Today I'm going to present top bioinformatics tools for drug discovery, sequencing analysis, NGS, and some of the summarized leading tools. So that is why the heading is top bioinformatics tools. And then it is also drug discovery and more. So here, for example, these days the most common things which are used are AlphaFold 2, 3, for which even the Nobel Prize have been awarded recently in last session. So this is like AlphaFold 2. And then there are some more things like molecular docking and virtual screenings. Then you can go for multiomics integration, CRISPR design. This is very demanding for gene editing. It's then pathogen surveillance. And finally, there are workflow for automation. This is here. Okay. So, for example, AlphaFold 2, which is these days highly authentic, and comparison to the previous softwares for prediction of protein structures, that is one of the finest software and highly accurate in terms of time taken. It is also quite fast. So AlphaFold is used to predict 3D structures of the proteins based on the primary structure of protein or amino acid sequences. It is crucial for structure-based drug design and better understanding of protein ligand interactions. So that is what you can hear. And this is one of the reference figure, which you could see how it works and what are the structures you could expect from AlphaFold. Then examples for the protein structure predictions. You could go AlphaFold 3.0, Rosetta Fold, ESM Fold. Then in the next step, it is multi-omics integration. By the way, more details have been presented about docking and structure prediction in my previous lectures also, which you could see in my like the previous presentation or lecture. Now in multiomics, this has been presented in one dedicated lecture in the previous lecture for 10 to 15 minutes, where you could recommend and you could see the slides with more details. So multiomics integration platforms, it generally combines genomics, transcriptomics, proteomics, and metabolomics data. It also offers the holistic view for disease mechanisms, understanding, and drug target predictions. Or you can also see phenotypic predictions. For example, Omicset, iCluster Plus, Use Galaxy is also here. Then this is the reference figure for explaining how to present or how to implement the multiomics integration pipeline. So this is one of the example. For example, here you could collect the genomic data, then go for transcriptomics data collection, proteome and metabolome data. Finally, you could combine them like data integration with computational tools, gene annotation and identification of candidate genes, functional analysis using reverse genetics. In this phase, some of those people who are not experienced in computational programming or any kind of coding, they normally face the problems, but I think it's quite easy if you could read a book about Unix command and awk commands that will be very simple to write some small codes as a layman, and there is no need to understand some big or complex coding. In case if you want to skip this step, then you need to follow the or use the like software package, big package, that is for example here Galaxy, that is also called usegalaxy.org. So this can be considered as one of these software package for integration of multiomics data. Next step is here molecular docking and virtual screening. So this could be seen here. So for proceeding towards molecular docking and virtual screening, I have one dedicated lecture for molecular docking, which basically presented the idea of how to develop your own software or how to use the other software and understand the basic principle behind molecular docking. And then it is virtual screening. So this overall heading, molecular docking and virtual screening, is meant to simulate how small molecules could bind to target proteins, and it speeds up the lead compound identification. And the examples softwares are AutoDock by RX, Schrödinger Suite, SwissDoc, and CBDoc2 and some more softwares. This is kind of pipeline for understanding the drug screening approach. Now, the next part is sequence analysis and target identification. So it's quite pure bioinformatics and easy things for which you don't need to learn any kind of coding or programming. So here you could identify the genes or proteins involved in these pathways, and it helps in as a pinpoint novel drug targets. And some of the noted softwares are Blast, Cluster W, Omega, Ensemble, David, and T-Coffee, and some more. Then this is an sketch to present the single cell TCG as a spatial cell transcriptomics, single cell multiomics, then single cell data integration, free single cell sequencing, then single cell CRISPR. Then you could go for high throughput screening, target identification, pharmacokinetic evaluation, and finally you could go for Swiss admit or admit properties identification, and then apply its applications are for single cell technologies in drug discovery for tumor treatment or tumor eradication program. Then some of the like the specific softwares, for example, or databases are, this is PDB for protein 3D protein structure, and then UniProt is for primary sequence of any known protein. So these two are the most common softwares, databases, which are required for before proceeding for protein drug target identification. Then next is, this is one of the example, if you want to use any kind of web servers for the software, then this is Swiss website where you could find a number of highly relevant softwares available in just one web server. So for example, this is SwissDoc, then Swiss parameter, then here Swiss target prediction. So you basically you could predict the target proteins for each specific drug. Then Swiss admit, if you want to predict the drug properties, use this software for this purpose. Then this is use Galaxy software. This is for multi-omics data integration purpose. So this can be used for any kind of NGS data set, whether it is genomics data, epigenomics data, or any related data, single cell genomics, single cell or spatial transcriptomics. This will be quite helpful. Then next is like other example, if somebody wants to go for sequence alignment, that this is one of the highly used wave server that is called multiple sequence alignment or MSA. This could be used for the protein sequence alignment or DNA sequence alignment, and it contains a list of softwares. For example, this is cluster omega or cluster W. Then there are other softwares, emboss, k-align, maft, then muscle 5, then mviews, t-coffee, wave prank. So these are the list of softwares for the purpose of sequence alignment, and this sequence could be of any types. So there are some more softwares, and here I have presented the summarized part for the most common softwares in the field of bioinformatics, drug screening, or docking purpose. So this is overall, and then there will be some more lectures on the related topics. So for today, this is sufficient. Thank you.